I think we'll get started. We'll probably have a few more people straggle in, but it is 9 o'clock, and welcome to Third Thursdays of our R&D meeting of the BC Wine Grape Council. Today we have a presentation on towards mitigating the impact of grapevine viruses in BC, and it's super important topic, so I want to get onto it right away. We have Dr. Jose Urbez-Torres and Dr. Tom Lowery presenting, and I'll just introduce them. Most of you probably know them, but I think it's important that that you understand what their background is and how phenomenal they are. Dr. Jose Ramon Urbez-Torres, and I don't know if I did a good job on that, but is a research scientist at Summerland Research and Development Center and serves as an adjunct professor in the biology department at UBCO. He received postgraduate master's degree in viticulture, enology, and wine marketing in 2001 from the International Social Science Council, and the degree of agricultural engineering in 2004 from the University of Valladolid.
Good.
I practiced that one. In Spain. Jose completed a PhD in plant pathology in 2009 in the plant pathology department at UC Davis. He has studied diseases of woody perennial crops, primarily grapevines, tree fruits, and nut crops since 1999. And his current research focuses on the development and implementation of sustainable management strategies for fungal, bacterial, and viral diseases of grapevines and tree fruits in Canada. He has served as president of the American Phytopathological Society and Pacific Division, and he is the current chair of the International Council for Grapevine Trunk Diseases. And Dr. Tom Lowery is a graduate of the University of Guelph, and he received his PhD from UBC Vancouver. He has nearly 25 years of research experience at the Summerland Research and Development Center on sustainable grape pest management, including chemical and biological controls, leafhopper anti-feedants, and the use of beneficial vineyard ground cover vegetation. He's also conducted research on the epidemiology and management of insect-borne plant diseases, including work with grapevine viruses and their vectors since 2011. He was an affiliate with Covey at Brock and an associate professor with UBCO. Tom has served on a number of BC Wine Grape Council research and development committees for many years, and I can attest to that, and produced the insect and mite chapter of the BC Production Guide for Grapes, and along with the accompanying photo guides for grapevine pests and for beneficial insects. I can't say enough about what these two men have done for the Wine Grape Council and the BC wine industry. Like their generosity and knowledge and insight has been invaluable, and I'll let them get on with it. Jose is first. Take it away.
Thank you very much. Okay, I'm gonna share my presentation. Hopefully it works. Let's go here. Everybody can see the presentation? All good? Yeah, okay. They can hear me. Well, thank you, Kathy, for the great introduction. Well, we wouldn't be, doing all this work without the great support you guys give to us. So, I mean, we are also very, very grateful to be working with such a supportive industry towards research. So, I think it's a good team. So today, we are going to talk about some of the work that we have been doing in the current CAP project on the management of grapevine viruses and their insect vectors. The first and most important, it's not just Tom and myself that we are talking today, but this project has been a huge multidisciplinary team, with Pat Bowen, Carl Bogdanov, Kevin Nasser, and Dr. Pujari, that is currently at Brock University, and he has spent here 3 years working with us. So what we are talking today is not just the work of 1 or 2 people, it's a whole team approach. And I believe that's why we have been able to achieve so much. Brief summary, I'm gonna just very brief introduction just to set the tone of the presentation about viruses in BC. I'm gonna focus today primarily on the work we have done on the effects on plant health, grape and wine quality. The work we have been doing in controlling grapevine viruses, primarily the roguing pilot programs we are having. And then Tom will go ahead then with identification and management of the insect vectors. So, importance of grapevine viruses in BC, I think at this time, it doesn't really need to be presented. We know that grapes can host many different viruses. Currently, there are more than 80 viruses that have been described on grapes. However, not all of them cause a significant impact. And among the ones that are really concerned, not only in BC, but also across Canada or even worldwide, it would be grapevine leaf roll disease. As we can see here, we probably at this stage, most of us know the main symptoms with this red of the leaves, green veins, and the rolling, very characteristic, the rolling of the leaves in the red varieties. And in white varieties, of course, we are not going to see this red pigmentation of the leaves, but we see the rolling, and usually we are seeing a chlorotic symptom of the leaves, but it's not as easy to identify a leaf roll on white varieties versus red varieties. And of course, a new emerging disease, which is grapevine red blotch disease. So far limited to North America. It has been reported only in Canada, United States, and Mexico. It hasn't been reported somewhere else. And still I have troubles to differentiate leaf roll from red blotch in the field. But one of the main things is these blotchy patches we are seeing on the leaves. And if we have only red blotch infection in the vines, these leaves are not going to roll. That's one of the main characteristics versus leaf roll. So previous work have identified the incidence of this disease in British Columbia. And as we can see from north to south of the valley with the field surveys we conducted, we see a significant increase on the incidence in the south part of the valley. Of course, that's the region with most of the acreage planted. And in that area, more than close to 70% of the vineyards that we surveyed were infected with leaf roll. Among the different leaf rolls that we identify, we have in BC all leaf roll 1, 2, 3, and 4. But as you can see in the different parts, north, central Okanagan, and south Okanagan, leaf roll 3 makes the most prevalent, is the one most spread. And in the south Okanagan, most of the infections, as we can see, more than 30% is leaf roll 3. This is important because we know also leaf roll 3 is the one causing the most impacts and damage on vines. So regarding the spread, this is just a snapshot of one of the plots we have in Merlot in the south Okanagan. We have seen through the years, we started, for example, in 2013, this block has a 25% infection. And through the years, we have been monitoring, all this infection has been increased, a significant rapid spread. And we have been going to, we went from 2013 to 2017, almost doubling the incidence of leaf roll vines infections. So in some blocks, in those, as Tom will talk later, where the insect vectors are present in a high incidence, we are seeing a very, very fast spread of the disease. Grapevine red blotch, fortunately, even though it's present in British Columbia, the surveys we have done indicates that the incidence is much lower. Surveys done in 2014 or 2015, we have approximately less than 2 or 3% of the incidence was so significantly lower compared to leaf roll. But one of the things is, as we see now on the effects they cause, red blotch has a tremendous impact on the plant health. And with this study, we demonstrate and we prove that it's most likely that red blotch has been introduced into British Columbia from material brought into the provinces probably mostly from California, where looks like it was first detected and it's most prevalent. And the indications of our studies indicate that. For example, in 2014, most of our blocks that we sampled were some of the oldest blocks in the Okanagan. Most of that material was sourced from Europe. We barely found any red blotch in that material. When we change our survey and we target material brought from the United States in much younger blocks, that's where, even though still the incidence is significantly lower, that's where we started to see more red blotch. So those are the facts that tell us that most likely it was imported. In terms of incidence, the blocks that have red blotch is much lower than leaf roll. We were talking, between 20 or 60% incidence with leaf roll in some vineyards. With red blotch, what we are seeing basically are incidents between 5 and 10% if the blocks we have been monitoring. As you can see here in the spread of red blotch in these two examples of Cabernet Franc, is very clear, and we were able to show that the plants infected were introduced. In this specific case, these plants were planted as replacement plants after a winter kill that happened in 2009. These plants came, in 2010, 2011, and replacing those dead for winter kill. So you can see, the basically a completely random spread through the block compared to leaf roll, where we see more, through the line in specific areas that is growing. In terms of the effects, we are going to start with leaf roll 3. This is a study we have done on Cabernet Franc, and just give you an example of some of the data from 2013 to 2016. And just the snapshot here basically is to observe that the main impact that we are seeing over and over every single year is in bricks. We can see here that the bricks are affected year after year. Let's see if this helps a bit here. Always, between 1.5 and even, 2 bricks less. Other things, for example, as like berry weight or number of berries per cluster, titratable acidity, pH, is not impacted every year, and it's going to be depending on the year. But again, when those components are impacted, is usually vines infected has a much lower versus vines that are impacted. So again, basically with leaf roll, we are seeing a constant decrease of bricks through the years. And also we are seeing, a slowing of the maturation process. So basically when we are getting to early September, mid-March, those vines infected with leaf roll, they are taking much longer to gain any accumulation of sugars. Regarding of some of the characters of the berries, so again, we can see that one of the things we see constantly in most of the years is a difference on the anthocyanins, with vines infected with leaf roll with a much less concentration of anthocyanins. Some of the other things we are talking, similar to other parameters before, is going to depend on the year. For example, in 2014, we had much less concentration of tartaric flavonoids, and even the seed condensed tannin. But that is going to be depending on the year. The thing we can see for sure is anthocyanins are lower most of the years. Regarding the wines we have been doing with leaf roll infected vines, these are Cabernet Franc wines in 2014 and 2015 that the group, of course, of Kat Bogdanov and Pat Bowen helped us to make. In blue, you can see the infected vines. In red, you can see the healthy vines. I'm not a wine expert, so bear with me. I describe these slides, but overall, we are seeing in Grapevine leaf roll infected vines, most of those wines are going towards a more vegetative flavors and aromas. We are losing this red fruit that is very desired in red varieties. And in Cabernet Franc, for example, in 2015, that was one of the hottest years actually that we have in the valley. We see that this separation is not so significant, but we still see some of these wines going towards that area of vegetative aroma. So the effects are really going to depend on the maturity of the fruit. So these effects on the wine, similar to the fruit, is really going to be a year dependent on that. I just have some recent data that we have been able to put together on wide varieties. So this is the effects of Leaf Roll 3 on Sauvignon Blanc in harvest 2019 and 2021. So overall, just to see here, I can say that compared to red varieties, we haven't seen in the case of Sauvignon Blanc in this particular block any significant difference on any components we are looking at. On red, you have infected. On green, you have healthy vines. Number of clusters, yield, berry mass, bricks, pH, TA. In some cases, we see a slightly decrease, on infected. In some years, a slightly decrease actually on the healthy, but there is no statistically significant difference on that. This is quite interesting, and see how the impact in this specific case in Sauvignon Blanc with Leaf Roll 3 is not as marked as with red varieties. This is probably because, as we know, white varieties are harvested earlier, as the requires for the bricks or the soluble solids is lower. So that could be one of the hypotheses that we are not seeing that. But based on this data, Leaf Roll appears not to have such a big impact on this variety. We have also Sauvignon, Chardonnay, and Semillon blocks that we are working, and we are processing all the data to make sure that this is not just an effect on only one white variety, but compared to other varieties that we plant in the valley. Similar pruning weights, slightly lower pruning weights, so it's a little bit less vigor on infected vines, but again, not significantly different. And bud hardiness, we didn't see any difference on bud hardiness, infected versus not infected. And I have to say that the bud hardiness also done on red varieties, we didn't see any significant difference on Cabernet Franc, Merlot, Cabernet Sauvignon when they are infected with leaf roll. So leaf roll doesn't appear to affect the bud hardiness. So moving to the effects of red blotch, this is a completely different story, and we have to say that red blotch is bad in any way. Red blotch has a tremendous significant impact, and we can see here with the work we did, and it has been published in 2020 by Pat Bowen. We can see how tremendous effects on yield. Yield can be reduced up to 40%, much lower number of clusters, 20% less clusters. Berries, are much larger, bigger berries with higher berries, of course. And you can see, in these 2 years, all significant values. Let me see why it's not working. Sorry, I'm trying to pass the slides, but significant impact in soluble solids, as you can see here, up to 4 bricks lower, so much bigger impact than leaf roll, where we usually see about 2 bricks lower. Tritable acidity and pH, again, all values are significantly impacted by red blotch. This is how the fruit looks, and there is no doubt, why we are seeing so healthy vines. This is a Cabernet Franc infected vine. So you can see how a severe infection of red blotch affect, the crop. Very, very dramatic in this case. In terms of the wines, in this case, we did wines in 2018 and 2019. We did wines with different types of infected fruit, from total healthy fruit, 0%, all the way to 5% infection, 10% infection, and 20% infection, and 100% infected fruit. So we can see here, of course, in 2018, all 100% infected fruit, very light color, vegetative flavor, even, and the same thing for 2019. But we can see also a trend that even up to with 20% of infected fruit, we can have some impact on some of these negative effects on the wines in wines coming from vineyards infected with red blotch. So again, this is not going to be year dependent, as we have been seeing, for example, with leaf roll. Every year, fruit infected with red blotch is going to have a dramatic effect also on the wine produced from those blocks. Another important thing, it has also a tremendous impact on bud hardiness, and we can see here, red blotch infected vines, how they have a significant decrease 2.5 degrees average in bad hardiness. So I don't know if in the positive way, maybe grapevines infected with red blotch in a year like this, where we have such a big cold snap, most of these vines, we have the buds dead and they can be easily identified. But based also on what discussion with Karl Bogdanov, there are very few things that can affect bad hardiness, but red blotch is one of them, very significant, and for that reason also important in our region. So finally, to finish my part of the presentation, I just wanted to talk a bit about the roguing that we have been pilot program that we have been doing. In this case with red blotch. This shows you a Cabernet Franc block I showed you before. This Cabernet Franc with about 11% infection in 2018. So we went to this block, flagged all these vines. All the vines, were tested. So we are talking about more than 3,800 vines. We wanted to make sure that the vines that needs to be removed is infected and is not caused by any other cause. So all the vines were tested and removed. So in 2019, we were able to go from 11.5% all the way just to 0.3%. So only 11 vines were infected. Some of these vines were because the flags were lost and they were still from 2018. So they are actually not new infections, but we can see how, we significantly reduce. And this was just removing the infected vine. In 2020, we have some new vines showing. In this case, a couple of new vines showing, but we still are way, way below the incidence, continue doing this roguing. And this roguing, as I mentioned, it was just doing the infected vine. Another block we have in 2018, in this case, in this block, we have only a 5% infection for red blotch. Similar, all the vines tested, flag in 2018. All these vines were removed. So we went down to 0.2%. 9 new vines appear, and in this case, a little bit higher in 2020. We can see 1.1%. Even though we removed these vines, we have 38 new vines. So we are seeing some spread, not as fast and rapid as leaf roll, but we can see here that even doing this roguing, we are seeing a spreading of some vines. It's very interesting to see, for example, that those vines came symptomatic in 2020 and not in 2019, meaning that if these healthy vines were infected in 2019, it's gonna take up to 2 years to see infection in those vines. We are not gonna see the newly infected vines infected the year after. We have shown that it's taking at least 2 years. Still, we have been significantly reducing the infection. And as I said, this is just removing the infected vines. So we know that now what we are seeing is when we remove one, infected vine, we remove this is the ideal, that you will have to remove all these adjacent vines to eliminate any potential spread. In our case, we only remove the infected vines, and what we, saw most frequently was just the adjacent vine was showing symptoms 2 years after. So is in most of the cases the adjacent vines for red blotch. So the last year, we recommend to the grower to remove not only the infected vines, but also the 2 adjacent vines. And we are hoping that with this, we were able to basically eliminate all the infection in these blocks. I had to mention also that since we observed that this pattern is exactly the adjacent vine to the one infected, I've been working with Tom Lowery. We are just trying to assess if, for example, nematodes could have a role on transmitting the virus. Is very unlikely because there is no reports of nematodes being vectors of these Gemini viruses. But I think it was just worth of a shot to work with Tom Forge to also try to eliminate any possibility just because of that, because exactly the vine next to it also gets infected. So this is all I have from my end. I can pass it to Tom. Tom, are you ready? I'm gonna stop sharing.
Okay. Want to continue? Yes. Oh. hopefully everyone can see this. I'm going to
Yeah.
put it on the slide format here. And everyone can hear me okay?
Yeah, you are good, Tom.
Good. Well, thank you very much. Great introduction. Thank you, Jose, for setting the table here for me to carry on more specifically with some information on the vectors, the spread of the grapevine viruses. So what we're going to look at here, vineyards that have some infection and primary infection. Typically, we see nursery material coming in that has leaf roll virus infection. This happens on the picture on the left to be a white variety. And you have then secondary spread of grapevine leaf roll virus through vectors, insect vectors, and you end up with spread into neighboring what were clean vines. So that's the background that I want you to keep in mind. We're responsible for bringing in infected nursery material, but then we have insect vectors that help spread it. A good source looking at what vectors we're talking about, paper in 2017, it was actually a chapter in a book on Herbach et al., vector transmission of grapevine leaf roll associated viruses. And he lists in this chapter a total of nearly 20 species that have now been shown to transmit leaf roll virus. You can basically assume that all of the mealybug and soft scale insects are able to transmit leaf roll viruses to a variable degree. The exception is leaf roll 2, and don't forget, these are actually different viruses, 1, 3, 4, and 2. There is no known vector for leaf roll 2. So we know there's 20, nearly 20 have been shown to transmit, but what do we have in BC? So we did quite a bit of work on this, looking at surveys, using molecular work, help from Ottawa. We have largely predominantly three that are responsible for the spread of leaf roll virus in our vineyards. There are a few other minor mealybug and soft scale, but they're quite rare. So what we have is grape mealybug, European fruit licanium scale, and cottony vine scale, which was formerly misidentified as cottony maple scale. So of those three, which is the most important vector? So in relation to the spread of leaf roll virus, the most important stage is the active crawler stage. So these are the small, you see the picture down here on the left of some crawlers caught on sticky tape, and they have the longest legs proportionate to the body size, and they're the active, they're very small. So they can be blown around even on the wind. They're the dispersal stage. So grape mealybug has 2 generations per year, so it produces crawlers twice a year, where the soft scale only have 1 generation. The rapid spread of leaf roll 3 is clearly linked to mealybug outbreaks. If you look in the South Okanagan, where we had the biggest problem with leaf roll virus, that's where the mealybug populations occur in the largest numbers. So they're pretty much restricted. We see a few of them for the first time. They're spreading a little bit farther north. We had some, recorded some from the station for the first time last year. But largely where the rapid spread of leaf roll virus occurs overlaps with where we see large numbers of grape mealybug. So that's the one we're mostly concerned about, shown down here on the bottom. And these adults, mealybugs, they do move around, and even the nymphs, they'll relocate quite a bit, where the soft scale tends to stay put a little bit more. So it seems to be mealybug are the problem. But we need to understand the biology of both mealybug and scale. So there's some differences that you need to look for. So mealybugs winter as eggs or crawlers in those cottony masses that you'll find under the bark. And as I mentioned, there's 2 generations a year. Soft scale winter is partly grown diaposing females under the bark. And here's a picture on the right that shows you, here's some wintering European fruit Lacanium scale. We've pulled back the bark. Often they're a bigger problem with scale than you realize. You have to get down and tear off some bark, and then you'll find all these, once you expose them, there can be large numbers hiding under the bark. So another difference too, you'll sometimes find cottony vine scale on the bottom about 2 feet of the previous year's growth. So they're not always on just the older woody material, the trunk and the cordon. They can be on the previous year's growth. The European fruit Lacanium scale, we just don't find it on the previous year's growth. Females produce hundreds of eggs. And I mentioned the crawler stage, that's really important. It's also important for timing some of the control measures that you try to get a handle on when those crawlers are dispersing. And of great importance, predators and parasitoids help regulate populations at various stages of scale and mealybug development. So here's what some of the things we want to consider. So now I've given you the background. We know what the leaf roll vectors are. How do we manage the virus? Well, obviously the first thing we want to look at is sourcing of clean nursery material. So starting off with clean material. Then we need to consider controlling the insect vectors. You might need to remove virus source material. And I'm going to touch on buffer zones, a little bit of work we've been doing on that. And then there's also involving testing, roguing, and perhaps block removal. So I'm going to focus on these 2 areas, control of insect vectors, and tell you a little bit about the work we've been doing on buffer zone, which really relates to removal of virus sources. So management of the vectors. So mealybug and soft scale, just a reminder that they don't really cause a great deal of direct feeding damage. It's very rare. If you have extremely high numbers of mealybug, I've seen a little bit of slowed growth with those. But the concern is with viruses, with the spread of virus. And so the threshold for direct damage, feeding damage, is extremely high. Conversely, the threshold for virus transmission is extremely low. So we want to maintain mealybug and scale as low numbers as possible. So we have basically four control measures that we can use. Late dormant oils, contact insecticides or oils that have to be directed against the crawlers. We have foliar systemic insecticides that can be targeted against the nymphs that have settled and are feeding on the leaves. And then, of course, we want to maintain predators and parasitoids as much as possible. So dormant oil, I see that more people are using this. We had a talk just recently, and Hans Buchler helped out with that. We were talking about leafhopper management, but dormant oils can be used to help manage diseases, as well as control of overwintering mealybug and scale. Veg oil crop oil is registered for control of soft scales. The other oils that you might be applying for disease control, they will also help control soft scale and mealybug. So need to apply them, of course, with high spray volume directed to the cordon and trunk for good coverage. And as I showed you that picture, it's less effective when there is a lot of loose bark on the trunk. We applied the oils in our trials using a recapture sprayer. It wasn't optimal, but it's the equipment that we had on hand. And I'm going to show you, we still got some pretty good control. So this was a preliminary trial. I've shown this before, comparing dormant oil, Movento, dormant oil plus Movento, control, and a malathion spray that's targeted to the crawlers. So here's the approximate times: dormant oil put on in early April, Malathion was put on around when the crawlers, so these peaks here, these are the crawlers that we're capturing on the sticky tape. Movento was put on before that period. So you can see here that with looking at the controls, the numbers that were nearly 100, we had a reduction in about 50% reduction with malathion. So not the best. Our timing might have been off a little bit. Pretty good control with all the other materials. If you look here, the combination of dormant oil and Movento, really no difference to Movento used alone. And then the dormant oil did a pretty good job. So it was down around 25 compared to nearly 100. So we had pretty good control with any of those options. So chemical control of mealybug and soft scale. So we've been working to correspond with chemical companies to determine the best available options. So we can do trials, but if there's no chance that the companies are going to pursue registration of these products, we're really trying to get them on side. We've been conducting lab and field evaluations of new products and also correlating numbers of mealybugs and soft scale with insecticide applications. And you've heard me appeal for collaborations from industry members that if you're willing to give us some background information on the sprays you've used, we're interested to look at the resulting effect on the mealybug and scale populations. And of course, refer to pest control products recommended for use on grapes in British Columbia, the BC Ministry of Agriculture website. But I wanted to get back to this, why we're looking at the effect of, or the correlation of mealybug and soft scale numbers with insecticide applications. With certain persistent materials, we've seen an effect on predators and parasitoids and mealybug and scale populations over a period of 2 to 3 years. So what we've seen happening is, and these are large outbreaks of mealybug and scale, but it's related to sprays of persistent insecticides for leafhopper control. So if you use some materials, for example, some of the earlier work with neonicotinoids, the first year you're reducing mealybug and scale a fair bit, but you're also having a tremendous impact on predators and parasitoids. The second year, you start to see a bounce back, a rebound in mealybug and scale populations because you're still having an effect on predators and parasitoids. And sometimes it takes up to 3 years, third year after the spray was applied before we start getting things back into balance. I wanted to bring this up because I wanted to remind industry members that if you have put on some of these persistent materials like neonics, to pay attention not just to the leafhopper population, because they will be controlled for at least the next year, but you might miss the increase in the mealybug and scale populations. Okay, I mentioned predators and parasitoids. So this is basically what we believe. It seems obvious. This is what's happening, why we're seeing a rebound, resurgence in mealybug and scale. Biological control. There are a number of predators and parasitoids that help keep mealybug and scale under control. There's some generalist ones that are feeding. This one here, it's actually a little lady beetle larvae called, and there's one that's called appropriately mealybug destroyer. So these little Stethorus beetles, and they'll feed on a number of things. They're generalists, but they really like mealybug and soft scale nymphs. Here's a picture down here, looks like a big potato, but that's actually a soft scale, and you can see the whole emergence holes where the parasitoids have come out. And this is the overwintering scale. A picture on the bottom right shows an emergence hole where this is a nymph of a scale, soft scale, on a leaf, and it was parasitized, and there's the parasite crawled out, chewed a hole out, and emerged. So there are a number of them, and I just wanted to give you a little bit of the work that my technician, Andrea Bronner's been doing. So she collected 1,209 leafhopper scale specimens and reared them out in the lab. And from those, if you look at the overwintering soft scale, so collection of these winter forms towards spring, and what she found was 6 different parasitoid species shown here in these photos, reared from these scale, 990 individual specimens from 42 adult scales. So not only a lot of species, but big numbers of them, even multiple where you get more than one parasitoid emerging from a scale. And 80% of the overwintered adult scales were parasitized. So there's big numbers of those. Also, if you look at the scale nymphs, so now we're looking at nymphs that are on the leaves, there's also a high parasitism rate. And this time, 13 different species of parasitoid from scale nymphs, 219 individual specimens, and here up to 67.5% of the nymphs were parasitized. So 80% of the wintering ones, 67.5% of the nymphs on the leaves. And it doesn't end with parasitoids. There's also, she found 2 species of flies, little tiny flies, Leucophis species. Leucophis, these ones are quite specific, where they're not so much generalists. They feed on the soft scale, feed on the eggs and emerging crawlers. So you can't really see them very well in here, but they're these little fly larvae, typical maggoty type fly larvae that crawl through these cottony masses, and they eat up the eggs and the emerging crawlers. So in 2019, 93% of the adult scale egg masses had up to 5 silver fly larvae inside them. So there's a high predation rate as well. I want to switch over now to the possible use of buffers. So what we did in a number of sites, we've mapped infections in new grape plantings over multiple years. We've calculated the distance between new infections and nearest possible virus source. So we've chosen sites, preferably where we had, demonstrated here on the left, we have infected a block that's highly infected with leaf roll virus. And we do all the testing to make sure that, it's leaf roll 3, it's one that will be transmitted by scale and mealybug. And then we go in over multiple years, and as indicated in the orange rectangles, these are positive infections. Ignore the green ones, that's indicating something else. But if you look at the distribution, we get over a number of years, this is the accumulated accumulated number of positive infections over time. So I'm showing you here just a representative section of one study plot. So what we found, we assessed the number of grape mealybug and soft scale vectors as well, present in the virus source blocks. And I have to mention here that the work, we set this out in an area that used to have large numbers of mealybugs and very rapid rate of spread. But just so happened that due to a number of factors, it was almost entirely soft scale rather than mealybug when we did this work. So what I'm going to show you is representative, or clearly representative for scale, but maybe not so much. We need to do a little more work where we have predominantly mealybug. So that's the setup. And what did we get out of that? Looking at all the sites over all the years, the distance from the nearest possible source of the virus, we ended up, when we plot this, we end up with this lovely curve, a high R-squared rate, so it correlates really well. And if you look at this, the results show that about 69% of the new infections occurred within 25 meters from the nearest source. Another 28% occurred up to less than under 50 meters. So we were only left with about 3%. There was only 1 or 2 at each of the 5 meter intervals past about 45 meters. So if you had a buffer of about 50 meters, in this case, what we've done so far, you would have eliminated about 93% of your new infections. So it's something growers might want to consider when they're putting in new plantings. Is it worthwhile establishing a bit of a buffer? Most useful if you're doing a series of plantings and you can leave a buffer and then replant it the next year, for example. But the effectiveness of the buffers, of course, is enhanced with testing and roguing, as Jose was talking about. It will eventually, you will get virus secondary spread within the new planting. So what we're talking about here is trying to maintain a healthy, uninfected block of new vines. The alternative to this buffering and trying to keep the plants clean is block removal and perhaps scheduled area-wide replanting. So it's a little more drastic move where you'd have to take out whole blocks, clean things up. We've done some work on red blotch virus vectors, and this is primarily the focus of the MSc research by Dieter Kahl. So Dieter just graduated recently, and he did lots of work looking at surveying for the pests. We built on the work that came out of California that suggested that the 3-cornered alfalfa tree hopper was a vector. We don't have 3-cornered alfalfa tree hopper, but we do have 2 species, buffalo tree hopper. And we also have a third, a close relative that wasn't very common. So we've not yet shown transmission to grapevines in greenhouse trials using the tree hoppers. Here's one shown here in the bottom left. But what we have shown is the capacity to transmit red blotch virus. And this was done using artificial diets and having the tree hoppers feed on infected source material and then feed into the artificial diet. And then we were testing the artificial diet for the presence of virus. This is due to the complex, these are persistent viruses. And so the virus has to circulate through the body of the vector. And in most cases, for most insects, it doesn't, that doesn't occur. It only happens, there needs to be a close association of the virus with the vector that allows for this passage from the gut into the body fluids and then into the salivary glands and then into the plant. So this evidence is strongly in support of the buffalo tree hopper being a vector. So how do we manage grapevine red blotch? It's spreading rapidly in other regions of North America, but as Jose pointed out, only slowly in BC. So management of the disease currently relies on planting of clean nursery material, diagnostic tests, and removal of infected vines. But we need to do a little bit more work on the tree hoppers, particularly the biology. We need to know when they're feeding on the grapes. We know they are, but grapes actually aren't a preferred host. In the lab, we have trouble maintaining adult tree hoppers on grapevines. They don't do well, and they'll die in a short period of time. But they clearly are feeding on grapes, and we need to figure out the timing for that if we're going to apply any chemical controls. No insecticides are currently registered for tree hopper control, but sprays for other pests, for example, leafhoppers, are likely to provide some control. But as I mentioned, timing could be an issue. So to summarize, leaf roll virus management, in addition to clean nursery stock and a reduction in virus sources, there is a need for effective management of the insect vectors. Vector control includes the use of oil sprays and insecticides targeted against the crawlers or the nymphs, combined with the preservation of parasitoids and other natural enemies. Numbers of European fruit Lacanian scale, cottony vine scale, and great mealybug vary between production regions and years, particularly in response to insecticide use. So as I mentioned, the sprays can cause big outbreaks in, depending on which materials you use, you can end up resulting in outbreaks of these vector populations. So we really want to be aware of that. We need additional study to assess and assist in the registration of some new insecticides and evaluate their effects on parasitism rates. So with that, I want to, first of all, acknowledge and thank the BC Wine Grape Council and the CGCN. And as Jose mentioned, it's been, the industry is a wonderful group to work with. We really appreciate all the financial support, but more importantly, all the collaborations and feedback that we get. You're a great group to work with. Help from the people at our research center as well, and most importantly, collaborating wineries and vineyards. So with that, thank you very much.
Thank you, Tom. So we're open now for Q&A, and we have a question from Carl right away for Tom. What is the latest information on the effects of Savanto Prime on beneficials?
We did a trial. We're working on Savanto Prime with Dave Neild, who's our minor use coordinator at Summerland at SURDC. So we're working with him on that. We don't have enough information yet that I would want to come out and state firmly. It definitely will have an effect, but what we're interested in, what we've seen so far that I can tell you, Savanto Prime does not seem to persist as long as some of the earlier ones. So we really, when we started with the first generation of neonics, we saw a persistence that went into the third year after planting. Savanto Prime does not appear to persist nearly as long. So we're not seeing that damage to extreme damage so far. But there's the immediate short-term effect, it has quite an impact. But we'll get you more information on that for next year, we hope.
Thanks, Tom. And Christine is asking, any insect models in the work?
Yes, we need models, and I think Elizabeth Volkovich was, I think I saw her signed on. We've been doing a little bit work on the timing. So what we need to do with these vectors, I did show there's 3 different vectors that we need to consider. And one of the problems, the crawler stages don't all emerge at the same time. So it is partly going to depend on which vector is predominant in your vineyard, but we need to have better indicators, models, and they could be phenological, where you're looking at growth stages, either of the vines or of, sometimes you can get very good weeds or some plants flowering, for example. I've done that before with some other pests that are an indicator of when the crawler is occurring, or even better, temperature-based models that you can time your sprays. Ideally, we're going to have those, a series of them, for best timing for leafhopper. There's various management stages, leaf removal, sprays, etc., as well as for the mealybug and scale.
Have you tested lime sulfur application efficacy on mealybug scale pest applied in the late dormant stage?
We haven't done that yet. It should be done. It's another one that we should look at. We've been working on the oils first, but the lime sulfur is something that should be looked at because people are interested in the effect. Of course, they'd love to get some disease control along with the vector control. The oils aren't as good for disease control. They do provide some disease control. And if you're going to choose a dormant oil, you might want to consider using the older oils that aren't as pure. Some of the new ones, they've been purified. I won't say too much. They're wonderful for summer horticultural oils, but they've actually removed all of the sulfur contents, or almost all of it removed. So they're highly purified. So some of the older oils that people might be familiar with, they actually still have, it's low, but they have more sulfur in them, and they will tend to be a little bit better. But generally, it's something that should be looked at, because it'd be great to get the disease control as well as the vector control.
Is there any need for fallowing before replant? Is there any indication that either virus remains in the soil?
There's no indication of, for example, you can prune in grapes and you do not get transmission on the pruners or any mechanical transmission. The transmission occurs either you bud on, you do it in the nursery, or it's with vectors. So there should not be, there's a very extremely low risk of any grafting or anything. However, there might be, where it's a concern and where the information some growers are hearing from California, they have mealybugs, that the vine mealybug that will survive on roots. A large proportion of the population goes on to roots. So they will actually persist underground on the roots, and so they can come back up if it's that remnants of the grapes. Our mealybugs don't go down into the ground, so it's less of a risk. So I don't think you're going to get any value from a fallow period, other than it's something you should be thinking about doing anyway for other reasons.
Yeah, I can add to Tom, as he said. So we know and we have shown that you can find both red blots and leaf roll in the roots, and we have found red blots also detected in the roots. However, there are very few studies, and as Tom mentioned, the probability of getting these roots that are left when you pull out the vines, I mean, it's almost impossible to pull entirely everything. It's going to be some roots left, but the probability to have those roots, grafted into the new plantings is very unlikely. Also, there are no studies how long the virus is able to survive in those remaining roots in the soil. So yeah, as Tom mentioned, is not really evidence that following is going to be any benefit for that.
Okay, we're coming up to our hard close here. Elizabeth has replied to Tom that they'll have some basic phenology models this year or next, but need to combine them with observations by Tom to use them on pest management. And a question for, I guess, both of you, what is a good plant host for parasitoid species for borders? And then I have one more question after that.
I leave that to Tom.
Well, I have to first of all mention that this is all quite new. There's been no previous study of the parasitoids of mealybug and scale conducted in BC. So it was quite surprising to see the numbers, and not only the population levels, but the numbers of species. Mm-hmm. So I can't say that I really up to speed yet on all the preferred hosts. Parasitoids in general like to have nectar sources, and it's sometimes not the obvious ones. Plants produce extra floral nectaries, some species of plants, so you could look that up and see if there's some, but generally the flowering plants. One of the things with these small little parasitoids, it's often the compound flowers with plants with smaller flowers than your big ones. And for example, some of the roses, they're not the old-fashioned roses, wild roses are better, but some of the newer ones aren't that great, and some of them don't even produce nectar. So Sorry, I don't really have great recommendations, but go with your general ones that are helpful for other parasitoids. A lot of these really small parasitoids that are very host specific, and they can go from the overwintering to the nymph, they may not feed a lot on anything else anyway. They might go from one host to another.
Mhm.
So we need to find out more about that.
So question for Jose, but I think it's probably also for Hans. From Carl, what can a grower request from a nursery as proof of the status and testing protocol at the point of purchase? Do we know of a source of certified virus-free material accessible to growers in BC?
Yeah. So of course the growers, when they purchase material, they should ask to the nursery they are purchasing material if they have any, if the material they are selling is certified virus-free, and what type of certification they have, or proof of the testing they are doing. If the nursery is following any of the Clean Plant programs, mostly of course in the United States at this moment, they could be able to provide with that. Regarding the material accessible to growers in BC, right now, to the best of my knowledge, Hans can correct me, because I believe we still don't have any official label of material certified virus-free in Canada or from Canadian nurseries, but Hans, correct me if I'm wrong. I think we are still not yet there, but growers can access material from certified Clean Plant programs in Washington State as well as in California. Growers can purchase from nurseries that participate in those Clean Plant programs, from the Foundation Plant Service or from the Washington State Clean Plant program. Those in California, they mostly follow the new protocol 2010 in certification, and they should have a list of all the diseases that they have been tested for. Some nurseries in California, all the nurseries are acquired, for example, in California with the Clean Plant program, they will receive plants free of viruses from the Foundation Plant Services. They will have their propagation blocks. And there's a number of, I think it's up to 10 different nurseries in California. They also follow the California Food Inspection Agency certification, which, they will have inspectors come in every X number of years to check those propagation blocks, if they are healthy and disease hasn't come into it. And they should be able to request all that information. So those are the best options we have. But I will leave to Hans. He can mention where we are at this point in Canada.
Yeah, just very briefly. We do have 3 certified nurseries through the CGCN, but one of them provides the market in Quebec and Nova Scotia exclusively, and to a large degree with hybrid material. And the other 2 are doing tissue culture propagation, and for now all on-rooted. But with the exception that there is Upper Canada growers in Ontario, they have actually bought out North Shore Grapevine Nursery, and that they are also going to be doing tabletop grafting. So the traditional type, and that they're looking into grafting of tissue culture material. But right now, if you want to order plants tomorrow that are certified through the CGCN, they will have to be on-rooted and tissue cultured for the coming year, for this year actually. And the one is located in Kelowna, the AgriForest. They do a little bit of grapevine tissue culture propagation, and they have access to all the G1 material of the CGCN repositories. So a lot of the popular clones that are planted in BC are available.
But Hans, is my understanding that right now the numbers, to a commercial level, if a grower needs to plant 5 acres, it's gonna be difficult to find that material right now here.
Well, with tissue culture, you can multiply very quickly, but it helps if you have a 1-year notice. Yeah. In order to ramp up production substantially. But if you're looking for a few thousand plants, I think that is fairly easy to do.
Thanks, Hans. And thank you, Jose and Tom. It was super interesting. I feel like we could do several sessions on this. So we have, the R&D committee has received quite a number of letters of intent for the next cycle of funding. So those of you who are on the call who are members of the R&D committee, and anyone within the industry is eligible to be part of that committee. If